Okay, let's dive right in. Welcome to This Explainer. Today, we're exploring the absolutely fascinating and honestly sometimes perilous world of the immune system. We usually think of our immune system as, our body's elite defenders, right? Tirelessly hunting down viruses and bacteria. But what actually happens when those defenders get a little too enthusiastic? Well, today we're breaking down hypersensitivity reactions. Basically, these are the biological mechanisms where our own immunity accidentally causes tissue injury instead of, preventing it.
So, here's our roadmap. We'll start with when immunity goes overboard. Then move through the four main types. Type I, immediate allergies. Type 2, cytotoxic reactions. Type 3, immune complexes. And finally, Type 4, delayed reactions. Starting with Section 1, when immunity goes overboard, understanding the baseline.
Put simply, hypersensitivity is an exaggerated immune response to an antigen. Normally, when your immune system is firing on all cylinders, it just neutrally clears out foreign invaders. No fuss. But in hypersensitivity, we see immunologic tissue injury. The wild part? The lesions and damage you experience aren't actually from the invading pathogen or allergen itself. No, they're produced by the violent interaction between an antigen and the products of your own immune response. Friendly fire, basically.
This brings us to the two major time buckets we drop these reactions into, all depending on their speed and duration. On one side, we've got immediate reactions. These happen incredibly fast. I'm talking seconds to minutes of encountering an antigen, and they're driven mostly by humoral antibodies just circulating in your blood. On the flip side, we have delayed reactions. These are a slow burn. They take about 24 to 48 hours to really develop, and they're primarily mediated by a cellular response rather than those free-floating antibodies.
Moving right along to Section 2, Type I, immediate allergies. Think of the mast cell as a tiny cellular landmine already loaded with preformed histamine granules. On the surface of that mast cell, you've got fixed IgE antibodies. When a free and foreign allergen, say a stray pollen grain or a peanut protein, comes along and binds to those antibodies, it triggers a wildly dramatic cellular event. It's the immune system's hair trigger alarm.
Let's break down exactly how this happens, step by step. First, the allergen enters a body that's already been sensitized. See, B cells have actually already produced specific IgE antibodies from a previous exposure. These IgE molecules firmly attach themselves to resident mast cells and basophils. Then comes the critical moment. The allergen cross-links those IgE antibodies. This essentially flips a mechanical switch, causing the mast cell to rapidly degranulate. It dumps powerful inflammatory mediators, chiefly histamine, straight into the surrounding tissue. And since histamine is a potent vasodilator and bronchoconstrictor, it rapidly expands blood vessels, causing massive edema and swelling, while simultaneously squeezing the airways tight, making it super difficult to breathe.
This exact biological mechanism is the culprit behind those common allergic reactions you're probably already familiar with. We're talking allergic rhinitis or hay fever, allergic asthma, and atopic dermatitis, which we commonly call eczema. But if this massive histamine release happens systemically, it leads to a truly life-threatening condition, systemic anaphylactic shock. The global vasodilation causes a catastrophic drop in blood pressure. That is exactly why we treat anaphylaxis with epinephrine. It rapidly activates receptors to force vasoconstriction, pulling that blood pressure back up and opening the airways to, quite literally, save the patient's life.
All right, on to Section 3, Type 2, cytotoxic reactions.
So, unlike Type 1, where antibodies react with those free-floating allergens, Type 2 is tissue-specific. In this scenario, your humoral antibodies, specifically the IgG and IgM classes, make a huge mistake. They bind directly to antigens that are physically attached to the surface of your own perfectly healthy cells. Your immune system suddenly flags these own body cells as being of foreign origin. This kicks off a deadly cascade that recruits macrophages, dendritic cells, and the complement system to come in and destroy your own tissue. It's a classic case of mistaken identity.
We can actually organize this friendly fire into three distinct sub-mechanisms. The first one targets blood cells. For instance, if you receive a mismatched blood transfusion, your antibodies will bind straight to the donor red blood cells and destroy them. The second mechanism targets specific tissue components. A really fascinating example here is Graves' disease, where an autoantibody binds directly to TSH receptors in the thyroid, driving hyperthyroidism. Or take myasthenia gravis, where antibodies block acetylcholine receptors in your muscles, resulting in severe weakness. Finally, the The third mechanism is antibody-dependent cell-mediated cytotoxicity, or ADCC. The body normally uses this to lyse or burst tumor cells and large parasites using targeted leukocytes.
Next up is Section 4, Type 3, Immune Complexes. The really crucial takeaway here is that, unlike Type 2, the antigens and antibodies in Type 3 are both just free-floating in the blood. They bump into each other and clump together, forming what we call an antigen-antibody immune complex. Now, normally, your body acts like a street sweeper and just clears this debris away. But when there's an overwhelming amount, these clumps travel freely through your circulation until they physically get stuck. Imagine clogged pipes. They wedge themselves into the tight basement membranes of your blood vessels, joints, and kidneys. Once stuck, they activate the complement system, unleashing reactive oxygen species and lysozymes. The surrounding tissue gets completely wrecked. And why? Not because it was directly targeted, but simply because it had the bad luck of being right next to this massive, localized inflammatory fire. Because these immune complexes circulate all over the body before lodging into small capillary beds, they tend to cause widespread, debilitating conditions. If they lodge in your joints, you get severe arthritis, with all that swelling and tenderness. If they get stuck in the tiny filters of your kidneys, it triggers glomerulonephritis. Another really serious clinical example is Goodpasture syndrome. It's a rare and potentially fatal autoimmune disease where these complexes get stuck and trigger severe destruction in both the lungs and the kidneys.
Moving right along to Section 5, Type IV, Delayed Reactions. Okay, pay close attention to this one, because Type 4 is definitely the odd one out of the group. Types 1, 2, and 3, they're all mediated by humoral antibodies, IgE, IgG, or IgM. But Type 4 completely abandons antibodies. It is mediated entirely by specific, sensitized T lymphocytes. Because it relies on physically mobilizing cells rather than just deploying free-floating proteins, it is purely a cell-mediated response.
Let's look at how this builds a much slower immune cascade. Step 1. An allergen enters the body and is essentially eaten by an antigen-presenting cell, typically a dendritic cell. The dendritic cell breaks it down and proudly presents a piece of it on its surface, using an MHC2 molecule. Step 2. A CD4-positive T helper cell comes along and recognizes this presented antigen. Step 3. This recognition triggers a massive proliferation of T cells that pump out powerful cytokines, particularly interferon gamma. And Step 4. This chemical signal activates heavily armed macrophages and CD8-positive cytotoxic T cells, directing them to aggressively destroy the target organ. This whole cellular recruitment process takes time, which is exactly why Type 4 reactions are delayed by 24 to 48 hours. It's a slow burn, but a destructive one.
This delayed, fiercely cellular mechanism is the exact driver behind quite a few conditions. It's the reason you develop contact dermatitis, that delayed blistering skin rash a day or two after brushing up against poison ivy or certain chemicals. But it's also the underlying mechanism for highly destructive autoimmune diseases. In multiple sclerosis, T cells actually orchestrate the breakdown of the protective myelin covering on your nerves. In Type 1 diabetes mellitus, this exact cell-mediated reaction specifically attacks and destroys the insulin-producing cells right in the pancreas. It is also the primary immune response mechanism we see in tuberculosis infections.
Finally, Section 6. Looking ahead to tolerance and therapies.
And this brings us to a truly provocative final thought for this explainer. We've seen how hypersensitivity, whether it's the histamine flood of Type 1, the friendly fire of Type 2, the clogged pipes of Type 3, or the slow cellular assault of Type 4, is essentially a massive failure of immune tolerance. The body basically loses its ability to distinguish self from non-self. Now, a healthy system uses mechanisms like clonal anergy, where self-reactive cells are forced into a state of unresponsiveness, or it utilizes suppressor T cells to actively shut down rogue immune attacks. So here's the big question. If we can fully decode exactly how our body loses this vital self-tolerance, can we engineer new therapeutic ways to artificially switch these suppressive pathways back on, quiet the alarm, and cure autoimmunity for good? It's literally the ultimate frontier in immunology. Thanks so much for joining me, and I hope this provided a clear, structured view into the beautifully complex, but sometimes chaotic machinery of your immune system.